Hi everybody. This week's lecture then is looking at muscle tissue. We've covered the skeletal system. We last week looked a little bit about some of the key muscles and where they're located. So we'll start to look a little bit closer at those muscles, what they do, how they do that. Through its ability to contract and relax, muscles are able to perform a number of key functions in the body. The first, probably most obvious one that we would all think of is the ability to produce movement. Um, this can be total body movements like walking, running, jumping, or what we call localized movements or smaller muscle movements. Writing with your pen. Hopefully, some of you are taking notes right now and doing that. So we can have what we call gross movements, which are the big muscle groups, or very fine movements, which involve small muscle groups. Um, in order to do this, it's reliant upon that interaction between the muscles and the bones. So as we know, muscles generally terminate via a tendon, which attaches to a bone at an insertion point and an origin that we discussed last time. And through contraction, they can transmit force to those bones, and movement can occur at joints. But we need to consider some other type of movement as well within there. So muscles are also responsible for moving substances around the body. The heart is made of cardiac muscle, and its contraction and relaxation pumps blood through the blood vessels. The walls of the blood vessels themselves have muscle tissue within them. They can dilate or constrict, which allows more or less blood to flow through them as required. And other muscle contractions can also move food, bile, enzymes, and all manner of other substances inside the body, and make sure that they get to the specific places where they need to get to. In addition to that, we've got the prevention of movement in some respects. So by regulating some of the internal organs, we have ring-shaped muscles called sphincters that, when contracted, stop things moving through them. When relaxed, allow movement through them. So we have those in the digestive system, in the urinary system, basically anywhere we've got an inlet or an outlet of a substance. Second major function then is the maintenance of posture. So holding us up in this upright position. Okay, again, this is relying on contraction, relaxation of key muscles, but we've got more of a sustained, longer contraction, which can help us, hold an upright position. Again, these are very, very different to some of the contractions we see in terms of big movements, because they're having to be held for much, much longer, and they probably have a much smaller range of motion. On from that, the stabilization of joints. I've already mentioned that in order to create movement, muscles cross joints, and the contractions of them can create muscular or skeletal movements and those big movements. But in doing so also, those muscles pull the bones closer together and ensure that the joints are a little bit more stable, okay? Reduce the ability of them to perhaps dislocate. You guys delivered initial sort of joint presentations last week, and we saw, in particular around the hip, we saw a lot of muscles crossing the hip and some very, very large muscles like gluteus maximus. And that helps add into this stability of the hip joint, which is really, really important. I believe what the group that did the hip mentioned it. Stability being crucial because we have to maintain this upright position, and we've got the whole upper body sitting on top of those hips. So that's really, really key. The final major function then of the muscles is to generate heat. So as muscle tissue contracts, it produces heat as a byproduct. To a certain extent, this is very, very useful. The heat that is released by the muscle can be used to maintain our normal body temperature. Body temperature sits in a very finite range, or a very small range. So we tend to be around about 37 degrees. and a shift, a very small shift either way, can be quite catastrophic to the human body. So if we get from that, I think it's 37.8 is roughly accurate. If we get up to about 40, then that is very seriously overheating. If we get below sort of 36, 35, then we are in danger of illness and injury on the other end of the spectrum. So the ability of the muscles to generate heat to keep us in that right area is very, very important. A couple of ways that's done. One, via friction. So as we'll see a little bit later on, muscles are constructed of fibers running parallel to each other. When those fibers contract and relax, they rub against each other, and friction is generated as a byproduct. And the other major way that heat is produced within the muscles is through the breakdown of energy. We'll not get too heavy into this because we will look at the energy systems during physiology of human performance in semester 2. But all sort of food substances and chemical substances in the body are made up of different molecules. They are joined together by chemical bonds. The breakdown of those chemical bonds are what we call exothermic reactions. So when the bond is broken to release energy, heat is also lost as a byproduct. And that heat helps maintain or increase core temperature. Now, as much as I said that can be very, very useful, particularly if we think about it in a cold environment, the first thing that tends to happen is we get these involuntary contractions of muscles to create shivering, because that's going to produce heat for us. If we're exercising in a hot environment, that can become problematic. Core temperature is already going to be higher because of the ambient temperature around us. And then the extra heat produced by the muscles can cause us to overheat quite quickly. So exercise in hot environments comes with those risks. The body obviously has ways of regulating its temperature. Again, something we'll talk about in semester 2 in physiology. But we also need to put things in place such as hydration and rest breaks. In order for muscles to perform those functions that we require, they have 4 key properties, and this enables it to perform the functions and also contribute to overall homeostasis within the body. The first of these properties is electrical excitability. This is the ability of a muscle to respond to electrical signals. And this is how we stimulate contraction in the body, interaction between the muscle tissue and the neural system. Okay, while often we may see the systems referred together as the neuromuscular system. Okay, the nerves have to have a role in stimulating muscle contraction. So central nervous system, i.e. the brain and the spine, sends action potentials out over a motor nerve that travels along a nerve to a muscle fiber, and that will trigger a contraction within the muscle. Okay, that next property that we're clearly linked to there is contractility. That's the ability of the muscle tissue to contract and generate a force once it is stimulated by that action potential. When the muscle contracts, it generates tension. Okay, that's the force. That is then passed on to its tendons or other attachment points and creates a pulling force on the origin and insertion. As we know from last time, the origin is fixed, so therefore that pulling force creates a movement at the end of the insertion, and we'll have flexion, extension, adduction, abduction, whatever it may be. That tension can also resist movement. We have different contraction types: isotonic contractions, isometric contractions. We'll talk about later on. They are both created by a contraction, a production of force, but movement is seen or not seen in a variety of different ways. Our third key property then is extensibility. That's the ability of a muscle to stretch without being damaged. So even in a stretched position, the muscle is going to be able to still generate force and produce a contraction. This is pretty important, to allow us to move through the full ranges of motion that we want, particularly in big body movements, sport and type actions. those muscles have to be able to get stretched quite far, particularly some muscles that are across two joints. If we think about the hamstring muscles across both the hip and the knee, if we are in a fully extended knee position, so legs straight, and then we also flex the hip, those hamstrings will get stretched out very, very far. Okay, and if they didn't have this property of extensibility, particularly if they're trying to generate force in those extended positions, that could be quite catastrophic for the muscle fibers, and we could get tears. Our final one is elasticity. Okay, the ability of that muscle tissue to return to its original length and shape after it's produced a contraction or after it's been extended and stretched out as far as it could. Okay, again, if that muscle didn't return to its original size and shape, it would lose some degree of functionality and it wouldn't be able to keep producing the sort of movements that we needed to. Much like we had when we looked at the bones in the skeleton, we can subclassify muscle tissue into different key classifications. The first of which is our skeletal muscle tissue. This is so called because it's the skeletal muscles that are primarily responsible for manipulating the skeleton and producing movement. Okay, you may also see these referred to as striped or striated muscles. It's because if we looked at them under a microscope, we can see alternating stripes of lighter and dark bands in the tissue, and we'll see that a little bit later on. Now, your skeletal muscles primarily work in a voluntary manner. The action of them is consciously controlled and then stimulated by the central nervous system. So we make the decision. So if we look, see the example on the screen there is the arm, biceps and triceps. We make the conscious decision to contract our biceps and perform a bicep curl. It doesn't happen automatically. Okay. Skeletal muscles can be controlled in an involuntary fashion. This would do with a sort of reflex movement, and that's to produce safety. Other certain areas, for example, the diaphragm would be classed as a skeletal muscle, but its action is involuntary because it's involved in that crucial process of breathing. The cardiac muscle is only found in the walls of the heart. Okay, cardiac muscle is also striated, so it's the same type of muscle tissue as we see in the skeletal muscles, but its action is involuntary. So we've got the repeated contraction and relaxation of the heart, which pumps the blood out towards the body. Okay. Its contraction is stimulated by the pacemaker within the heart, so it creates its own electrical stimulation, which controls that automatic rhythm of the heart. That can be manipulated by hormones and neurotransmitters released via our more central nervous system. Again, something we'll cover in a lot more detail in the physiology module in the second semester. One key factor really though that differentiates it from the striated muscles we find in the skeletal muscle tissue is that it doesn't fatigue. If you think about it, say a resting heart rate of around 70 beats per minute, your heart repeats that 70 times every minute of every hour of every day for your entire life. If you attempted to do 70 bicep curls every minute for several minutes, you probably wouldn't get through the first one or two. Our third type of muscle tissue then is smooth muscle tissue. Okay, this is found in the walls of hollow internal structures, so blood vessels, airways, and most of the organs involved in our digestive and urinary systems. We also find it within the skin and to control hair follicles and the movement of the hair, the ability of the hair to stand up to trap heat when we need it, and in the eyes to control dilation of our pupils. Called smooth because unlike striated under the microscope, it doesn't have those dark bands running through it. It has a smooth appearance. The action of this muscle tissue is involuntary. Okay, so it is run by the autonomic nervous system. So no conscious thought is required. We don't think about dilating our blood vessels. Okay, we don't think about ensuring food moves through our digestive system. Um, but the actions are regulated by other aspects of the nervous system and also the endocrine system, which releases hormones and, for example, can cause dilation of blood vessels to increase blood flow to certain key areas. From this point onwards, we are only going to be talking about skeletal muscles, okay? Because we're interested in the study of sport and exercise, it's these muscles and their ability to cause body movements that is going to be very, very important. So, although skeletal muscles can vary in size, shape, and arrangement, they all really consist of hundreds up to thousands of individual fibers packaged together to make a muscle. Key within there is also the layout of connective tissue. And then this arrangement of fibers in connective tissue is really, really important to then further understand how the generation of force can create movement. The connective tissues, first of all, are what we call a fascia. Okay? Within the muscles, we have various layers of those. So the sort of deep fascia is a dense connective tissue that lines the body and the limbs and holds muscles together. So various different muscles. Okay. They then extend from the deep fascia into individual skeletal muscles, and there are three layers of that. We've got the epimysium, which is the outermost layer of the fascia, and this encircles the whole muscle. Okay, so on the diagram on your screen now, if you look towards the right-hand side, where we've got an entire muscle together, right around the outside of this muscle is our epimysium, okay, deep fascia. We then have the perimysium. Okay, the perimysium is around groups of muscle fibers. So our muscle fibers, and you can see that cross section, you know, pink circles with smaller red circles inside. These are our bundles of muscle fibers. So from anywhere, depending on the size of the muscle, a bundle of fibers may be 10 fibers together, up to potentially 100 fibers together. And these bundles are called fascicles. Okay. The perimysium surrounds the fascicles and attaches those separate fascicles together, so the bundles aren't independent from each other. Within those fascicles, then, in between individual fibers, we have the endomysium. So this is like the within level of connective tissue. This again attaches individual fibers together so that they're not then moving independently. These layers of connective tissue are continuous. They run throughout the whole muscle. And it's also, they combine together to contribute to the overall muscle stability, structure, and strength. So all those three levels of connective tissue, endomysium, sort of within the fascicle, around the fibers, perimysium around the fascicles, so the bundles of fibers together, and the epimysium around the outside of the entire body, the outside of the entire muscle. They'll come together and join together into the tendon. Okay, so this helps to ensure that a muscular contraction is a whole muscle movement. Okay, we don't have, if there was just a few fibers contracting on their own and they weren't anchored to the rest of the muscle via connective tissue, we wouldn't get that contraction and shortening of a muscle on the whole. So this is a really, really crucial structure. Now, if we look sort of at this extended, we've got a single muscle fiber extending out of the fascicle, and we can see that there. Okay, excuse me. The fiber itself then has smaller myofibrils within it, all surrounded by what's called sarcoplasm. Okay, the sarcoplasm is a sort of it's a globular substance within which a lot of the essential fuels for muscles are found. So the energy that we use for contraction is within the sarcoplasm. It's a non-contractile element. It is outside of these myofibrils within which contraction is going to be created. And we also have in there myoglobin, which is an important oxygen store for within the muscles. And again, that's going to contribute to energy. Okay, so we will go on now to look at this smaller microscopic structure of the myofibrils. The myofibrils then are like tiny little threads running through an individual muscle fiber. This is where we see the striations or the stripes that give sort of skeletal muscle that alternative name, striated or striped muscle. So you can see on the diagram there the darker and lighter bands repeating along the length of the myofibril. It's also at this level where muscular force is produced. And again, at this level where we see differences caused by muscular atrophy, so the growth of muscle, or the decrease in size of muscle, or muscular hypertrophy, which is the growth in muscle. So that happens at a myofibrillar level. Each individual muscle fiber will have hundreds of these smaller threads running through it. And the myofibril is made up of sarcomeres. These are sections which repeat end on end along the full length of the myofibril. And it is at that tiny level of the sarcomere that muscular contraction is really triggered. Okay, and we can see a very, very close-up shot there where we see the sort of black actin filaments overlapping with the reddy brown myosin filaments in the middle. For those of you who've studied muscles before and done sliding filament theory, it's at this level where that occurs. Okay, we will take a closer look at the sarcomere on the next slide, but we're not going to get into sliding filament theory yet. That's going to be done in a week or two, most likely. So as way of a recap, at the very top of this diagram, we can see a single muscle fiber. Remember, there are hundreds, possibly thousands of these in each skeletal muscle, depending on the size of the individual muscle. We take a cross section of that muscle fiber, and we can see, tens up to 100 individual myofibrils running along the length of that muscle fiber. The myofibril then is arranged into distinct sections called the sarcomere, okay, individual compartments. Those compartments are made up of much smaller protein filaments. The thin filament called actin, the thick filament called myosin. Okay, and the arrangement of these is quite crucial. So these protein filaments do not run through the entire length of the muscle. They are much shorter and arranged into these compartments, this sarcomere formation, which, I like to imagine as like trains of a car. They're all attached to each other, they're all working together, but they are distinctly separate. Okay. The picture at the very bottom then shows you the arrangement of a sarcomere. Okay, and we've got those individual protein filaments within there. We've got what we call the contractile filaments, being the actin and the myosin, and we've got regulatory filaments and structural filaments, which we see as titin being a key one of those. Okay, so the overlapping nature of these thin and thick filaments is important, and it's not static. The less they overlap, the more extended the muscle will be. So when it's being stretched out, the more they overlap, the more contracted the muscle is. Okay, so that's when we see sort of force production occurring. And the mechanism for that contraction and extension is, again, what I mentioned just before, the sliding filament theory of muscular contraction that we will look at another time. The sarcomere and the layout of the sarcomere can be described by certain bands which indicate the position and the degree of overlap of some of those filaments. So we've got that darker middle band is the A band. That is the entire length of the thick filament, the myosin. We've got the I band, which tends to be lighter in colour. That is the distance between two adjacent thick filaments. Then we have the sort of H zone. The H zone is the distance between two opposite filaments within a single sarcomere. Again, this is stuff that'll make way more sense when we look at the actual contraction. So as the muscle shortens, the H zone gets smaller, potentially even completely disappears, but the A band will never change. Okay, the M line is sort of a middle point of the sarcomere. Okay, opposite thin filaments move towards the M line and potentially cross it. The Z disc is a fixed area. It passes through the sort of center of the I band, and that is the area that distinguishes one sarcomere from the next adjoining sarcomere. Okay, and again, we will come back to this structural layout and look at the actual mechanics of muscular contraction and force production when we look specifically at the sliding filament theory. The picture on screen then just shows you that microscopic image of a skeletal muscle. So you can see why it has the striated name. We can see these faint black lines running through the red muscle tissue. The white lines, I would add, that's connective tissue, so that might be tendons separating two sections of a muscle. This is meant to be a moving picture. Unfortunately, it's not moving today, but you would see the shortening and lengthening repeatedly of this muscle as it contracts and relaxes. But you can find that online if you're interested to take a look. Muscles can be grouped based upon the arrangement of the fascicles within it. So remember, fascicles are those bundles of muscle fibers, and we have a number of those bundles to form the overall muscle. The way they are laid out within the muscle makes a difference to sort of the range of motion or the strength that can be produced, which we'll come back to after we look at those individual groups. So the first group we can have is a convergent muscle, or sometimes called a triangular muscle. Here, the origin of the muscle is very, very broad. The fascicles, all coming from those origins, then tend to converge together into a singular, much smaller insertion point. Okay, the example we have here is pectoralis major. So if we look at the origin, we have that coming all the way up the sternum and also along a good portion of the clavicle or collarbone. The fibers then all come, or to use the term there, all converge together into a much smaller single insertion point on the humerus. So this results in a very large number of muscle fibers pulling together. Our second muscle form on this page here is the fusiform muscle. The fascicles here are almost parallel, and they terminate in sort of quite flat tendons. The muscles taper together towards those tendons, and they give this large sort of belly of the muscle within the middle. Okay, and like I say, they taper together, so the ends of the muscle are both thinner than the major belly of the muscle. But there is another muscle form similar to this called parallel, where we don't have that muscular belly and we get a much longer, thinner muscle where the fibers run completely parallel to one another. And sartorius in the upper thigh would be an example of that. We also have pennate muscles. In a pennate muscle, the fascicles are fairly short in relation to the overall length of the muscle. The tendon extends near the entire length of the muscle, generally through the middle, and those fascicles come off obliquely, so diagonally from the middle tendon. So the arrangement of the fibers in relation to the tendon is like a feather. So we see here the black line through the middle of that feather would be the tendon. The sort of the feather-like substances coming off the sides would be our muscle fibers coming off diagonally to attachment points at the other edge. Okay, so again, the key point here being a fairly short fascicle length, but what we do end up with is a lot of muscle fibers packed into a muscle because they are going up the full length of the tendon. And the pennate muscle can then be subdivided into 3 further types. We have unipennate muscles, where the fascicles run down just one side of the tendon. The example here given on the thing is a forearm muscle, extensor digitorum longus. We then have bipennate muscles. Tendon runs through the middle of the muscle and the fascicles come off both sides, like we saw with the feather on the previous picture. Rectus femoris, one of the major quadriceps muscles, is an example of a bipennate muscle. And then we also have multipennate muscles. So we have several tendons, each with fascicles coming off obliquely, okay, in multiple different directions. So a key example we have of this is the deltoid. So we mentioned last week when we were looking at positions of some key muscles, the deltoid having 3 major sections, anterior, posterior, medial. That's because there are 3 tendons coming off in different directions, inserting at the humerus. Each of those tendons has a large number of muscle fascicles coming off it in this sort of diagonal fashion, and the result being we have a very, very large amount of muscle fibers present in this individual muscle, and we can also create movement in several different directions. Now, the arrangement of those fascicles, whether they be convergent, parallel, fusiform, pennate, that makes a difference to the strength and power that a muscle can exert, and also the range of motion that can be produced from a muscle. So when a muscle contracts, it can shorten to about 70% of its resting length. So this makes sense that the longer muscles have a much bigger range of motion. So we mentioned on one of the previous slides, sartorius is a parallel muscle where we have a very long muscle fibers running parallel to one another. So we get a big range of motion within that. However, it means there can be less fibers, so it doesn't create as much strength. The exertion of strength and power from a muscle depends on the total number of cells in that muscle. And by cells, we can interchange that word with fibers. So the greater the number of muscle fibers within an individual muscle, the bigger the expression of strength and power can be. So by that respect, our bipennate and multipennate muscles pack in the most fibers, but those fibers are shorter in length. So they can exert a much greater deal of force, but move through a smaller range of motion. Okay, so we basically have this trade-off: greater force production or greater range of motion, and there's a compromise there depending on what's required at specific joints within the body. Now, remember this microscopic image of the muscle when we look at it closer. Don't worry, we're not going to get into all the terminology again. But one of the key things, remember, was individual muscle fibers within the muscle. However, what we do need to recognize is that not all muscle fibers are the same. And that's both in terms of their composition, what sort of fuels and substances they have within them, and also their function. So muscle fibers will vary in the amount of myoglobin they have. Myoglobin, remember, being a source or store of oxygen within the muscle fiber. And it's also the sort of protein that gives them their red color. They will also vary in the sort of speed at which they contract and relax. They'll have variance within their metabolic abilities, and definitely differences in the amount of force they can produce. So, based upon several structural and functional characteristics, we can subdivide our muscle fibers into three main groups. Um, I would imagine that you've all heard of fast and slow twitch fibers before. Um, we need to further look at that. So we have what we're going to call slow oxidative fibers. We have fast oxidative fibers and fast glycolytic fibers. And that's what we're going to go into over the next few slides. And it's also what we will focus on quite a bit during the in-college session later this week. Now, as I mentioned, the characteristics that everyone's probably familiar with are slow twitch muscle fibers and fast twitch muscle fibers. Slow twitch being the fibers that are more suited to endurance-based activities, fast twitch fibers being the ones that are more suited to our faster, more explosive speed and power activities. Okay, that division based on major characteristics, right? In truth, when you look at those characteristics, we get these three divisions. Okay, we have our slow oxidative fibers, also known as type 1 fibers. That's what we probably more traditionally just knew as slow twitch muscle fibers. Then our fast twitch muscle fibers are split into two categories. Fast oxidative, which is type 2A, fast glycolytic, which is type 2B. Now, this division is based on these two major characteristics. The first being our speed of contraction, fibers that either contract slowly or contract quickly. So that gives us the slow versus fast division. Then we also have the major characteristics of which energy system pathway primarily powers those fibers. So it's not that a muscle fiber is only aerobic or only anaerobic, but they are better suited to one or the other. Okay, we're not going to get too deep into this because energy systems and energy pathways are a semester 2 subject in physiology performance. But hopefully we have this underlying knowledge of aerobic versus anaerobic activity. Aerobic being energy systems that use oxygen, and as a result of that, they are slower to act and can only power exercise of a lower intensity, but can do a longer duration. So this is what we call oxidative energy. That's aerobic. Or glycolytic energy. This is our anaerobic activity. Okay, energy can be produced at a much faster rate, but these systems fatigue more quickly. Okay, so they can't be done for as long a duration. So we have slow oxidative, where muscles contract slowly. They use aerobic energy, and as such can do it for a long time, but at a low intensity. We have fast oxidative, where the speed of contraction is still fast, but they'll still rely primarily on the aerobic system. So they can't do it such a high intensity. And fast glycolytic, okay, fast contraction, high powered energy, but can't do this for as long a duration. We'll take a brief look at each of those fiber types separately. So we'll go with type 1, then we'll go type 2B, being that they are at the far ends of the spectrum from one another in terms of their characteristics. And then we'll speak briefly about type 2A at the end, because they are, as it says on there, intermediate fibers, okay, kind of in between the two others. So our slow oxidative fibers, type 1, are the smallest fibers in diameter and as such the least powerful type of muscle fiber. Okay, cross-sectional area of a muscle or diameter is directly related to the force production capability. Okay, the wider those fibers are, or they are wider generally because they have got more of those contractile protein filaments within them, more actin, more myosin, and as a result, they can create more force. So the smaller diameter fibers cannot produce as much force. So that's our slow oxidative fibers. They appear dark red when looked at under a microscope. This is because they've got large amounts of this protein myoglobin, which can hold on to a store of oxygen in the muscle. They also have very, very dense capillary networks. So the delivery of blood to them is much, much richer as well. Okay, and the combination of those two things gives them this red appearance. They have lots of large mitochondria. Again, people who have studied energy systems before will hopefully remember this cell, mitochondria, often referred to in some texts as the powerhouse of the cell. It is within mitochondria where aerobic energy production takes place. So the more mitochondria you have and the bigger they are, the more oxidative energy can be produced. They are slow to contract. This is because the key enzyme within those fibers that breaks down ATP, our source of energy, works at a fairly slower pace. Okay, so the breakdown of energy and then the resultant movement of the muscle fibers is not as quick as is in the fast fibers. So slow contraction speed, and they, as a result, can't produce their peak force as quickly as other fiber types can. They are, however, very, very resistant to fatigue. So they can produce prolonged contractions and sustain them potentially up for a number of hours. So these slow twitch and fatigue resistant fibers are perfect for low intensity, long duration activity. Okay, this can be as simple as the maintenance of posture. You can sit upright for a very, very long period of time, and it's going to be slow oxidative fibers that allow you to do that. Or endurance-based activities, jogging, running a marathon. You can run, if you're sufficiently trained for it, for 2, 3, 4 hours at a time. Our fast glycolytic fibers then, type 2b, at the very, very far end of the spectrum, are our largest fibers in diameter. Right? They've got the highest number of myofibrils within the individual fibers, and as such, they can generate the most powerful contractions. The flip side of that is they're going to have a low myoglobin content and a much smaller density of capillaries feeding them with blood and less mitochondria in them as well. Right? As a result, they have this white appearance as opposed to the dark red in our slow fibers. And it also makes them less able to generate aerobic energy. Okay, they do, however, have much higher storage amounts of glycogen, and therefore they're going to make their energy mainly from glycolysis, which is one of our primary anaerobic energy systems. The speed of ATP breakdown within the fast twitch fibers is much, much quicker. So as a result, they're able to generate force at a much quicker rate as well. So they're really well adapted for intense anaerobic movements where they are only short in duration but require faster and much more forceful contractions. Okay, so we're thinking about things like weightlifting, throwing, jumping. And the downside is they do fatigue fairly quickly. They as well, though, are capable of great changes in strength and force production as a result of training. In the middle, between those two ends of the spectrum, then, we have our fast oxidative fibers, our type 2A fibers. So they're intermediate fibers in many respects. Their size is somewhere in between our type 1 and our type 2B fibers. They still have large amounts of myoglobin, still have good capillary supply, so they have that red appearance. They can generate fairly considerable amount of energy via the aerobic systems, which gives them a fairly high resistance to fatigue, but they still also have a fairly high level of glycogen stored within the fibers too. So they can produce energy via anaerobic glycolysis also. And they've still got that fast contraction time, but again, they will fatigue slightly more quickly than our slow oxidative fibers. So it's kind of a best of both worlds situation with our type 2A. Okay, they've got a good endurance capacity, they've got a decent force output, so these fibers can suit both types of activity, both endurance and high intensity. Obviously, not as good at endurance as our type 1 fibers, not as good as high power force output as our type 2B, but a fairly good capacity for each. Generally speaking, your skeletal muscles will be a mixture of all three fibers, probably about half of which are slow oxidative fibers, the rest differentiated between the two types of faster fibers. The proportions do vary though, depending upon the specific action of the muscle, depending upon that individual person's training, and also genetic factors. So, for example, the major postural muscles of the neck, the back, and in the legs have a much higher proportion of our slow oxidative fibers. That's because we need those muscles to be able to work for extremely long periods of time without fatigue in order to maintain posture. Muscles of the shoulders and the arms, in contrast to that, aren't as constantly active, work much more intermittently, and probably produce much larger amounts of tension during activities such as lifting, throwing, whatever else you may need to do on a day-to-day basis. So those muscles have a high proportion of our fast glycolytic fibers. Some of our leg muscles, for example, which is why I've highlighted the calf on the picture here, gastrocnemius soleus. Now, they have a much more varied role within the body. So these muscles in the leg have to support the body, but they're also used repeatedly for walking and running. If you think about it, in order to propel our body off the ground while walking or more forcefully while running, there's a much higher force output required. So they have to have a really good variety of muscle fibers within them. Within those muscles, the fibers are grouped together into little functional components called motor units. A motor unit is a number of fibers distributed around the muscle, stimulated by a given nerve. Okay. Those motor units are recruited in a specific order depending on the task at hand. So, for example, if we need just a weak contraction, we'll recruit the slow oxidative motor units. So we'll just use the slow fibers, okay, because we need a less forceful contraction, but we maybe need to do it for a more prolonged time period. If more force is needed, we'll then recruit the fast oxidative fibers. However, then if we need maximal force, we'll begin to recruit to the motor units that have the fast glycolytic fibers. Okay? So we're recruiting them in order depending upon the task required at hand. Okay, like I say, training will have an effect. As muscle fibers sort of develop. However, in the main, the amount of each individual fiber we have will be dictated by genetics. Okay, we each have a predominant fiber type. If you are predominantly a slow twitch fiber individual, you can train your fast activities, you can train the glycolytic components, the anaerobic systems, and get better at them, but you will never be a champion in the 100-meter sprint. All right. Conversely, if you have a much more predominant makeup of fast glycolytic fibers, whilst you can certainly train to improve your endurance capacity, you're never going to be an elite marathon runner, right? We have sort of genetically based predispositions to certain sports, and everyone can probably have an idea as to how they are, how you respond to training. If you respond very, very well to endurance-based training, you can assume that you have a greater predominance of slow twitch fibers. If you respond very, very well to strength and power training and kind of adapt fairly quickly to that, you might assume you can have more fast glycolytic fibers. These things can be tested via muscle biopsies, where a researcher will take a chunk of muscle out and perform tests in a lab setting to see which fiber type is there. It's not really commonly done. It would only be done if you were a participant in a research study within a university, probably looking at fiber type for any different sort of number of reasons. Other than that, it's kind of just based upon your own individual knowledge about responses to training. A couple more areas to cover before we finish for today. Starting to come around towards more functional application of muscles in movement. And again, these last two discussion areas we're going to look at will be covered in a lot of depth, applied to real movements in class later this week. So the first one being types of muscular contraction. So contraction is the ability of a muscle to create force, right? And it's through this creation of force that movement of the body and movement of external objects by the body is created. We have two major types of contraction, these being isotonic and isometric. All right. Isotonic is a contraction within the muscle where there is a change in length of the muscle. And there are two types of isotonic contraction. The first being concentric isotonic contraction. This is where a muscle shortens and as such pulls on another structure, generally a tendon initially, to produce some sort of movement. And this will often reduce the angle of the joint. So for example, our biceps brachii shortens during concentric contraction, pulls on the tendon then pulls on the radius. We get flexion of the elbow as a result. Now, if we imagine that as in the bicep curl exercise, okay, lifting up our dumbbell. So our concentric contraction of biceps brachii produces the force required to cause flexion of the elbow and lift the dumbbell up, right? If we are then to lower that dumbbell back down, it is not a case of we just relax the biceps, okay? If we do that, the dumbbell falls in an uncontrolled fashion. It's probably way too fast, and as we result, or as we return to the anatomical position or full extension of the elbow, it'll probably be done at quite a high velocity, and this could force the elbow into hyperextension and damage our structures. So we need to lower it under control. And if we are to control that lowering, some degree of force has to be produced. So to do it under control, the same muscle, biceps brachii, has to continue to create tension, but gradually get longer. Okay? This is what we call an eccentric isotonic contraction. So this is still a contraction of the muscle. It is still generating force, but it is actually getting longer. All right, and it's eccentric contractions that we tend to find when we are having to produce some sort of controlled descent. So after the initial lifting phase of any weightlifting exercise, we have to lower that weight under control, and that is done by the same muscles that lifting it concentrically have to lower it eccentrically. All right. Couple of key points to bear in mind about eccentric contraction. First one, and this is due to something called the length tension relationship. we are able to generate more force eccentrically than we can concentrically. You know, anyone that lifts weights will probably find that you tend to fail on a lift trying to lift the weight back up. If we take the squat, for example, you take the bar out of the racks, you don't fail there. You get to the bottom of your squat, and then you either get stuck right at the bottom or you get part way back up and you fail. So you can control the descent, you can lower that heavy weight eccentrically and get to the bottom, but you don't have enough force to get it back up concentrically. So we can deal with a load eccentrically that we are unable to deal with concentrically. All right, we'll come back to that when we look at sliding filament theory and hopefully try to explain that. But it does govern a certain training method. You can train just the negative aspect of a lift to try and overcome a plateau in force production. In addition, when we get large amounts of eccentric work, we are probably going to get greater amount of muscle damage and a greater amount of muscle soreness as a result. Our next type of muscular contraction is isometric. These are very, very important during stabilization and posture control. Isometric contraction generates tension without any shortening of the muscle. Okay. So let's say, for example, if you're performing a plank, you know, we are generating huge amounts of force through the core muscles, transversus abdominis, being one that we mentioned last week. However, there's no visible movement occurring. The origin and insertion are not getting any closer together. Okay. Again, important in many day-to-day activities where we require posture. It can also be seen where we just can't generate enough force to overcome an external object. You know, if we go back to that squat idea, when you fail, let's say you unrack the bar, you go through the eccentric phase, get to the bottom of the squat, you begin the concentric phase, you get part way up, and it doesn't normally result in sudden catastrophic failure. We'll get a period where there's no movement occurring, isometric, we get stuck, we can maintain that isometric contraction for a little while, and then we maybe fatigue and fail. That's one of the major downsides of isometrics. We can fatigue quite quickly, and we also get a massive increase in blood pressure as a result. So it can be actually a useful training modality to implement into a program, because we get fairly high force production through isometrics, but it can be dangerous for certain population groups. A final contraction type is included on here, isokinetic contraction. Don't tend to group this as one of the sort of real ways that muscles can contract, because isokinetic can only be created in artificial situations. So an isokinetic contraction is a contraction where the speed of movement is constant. So we need to use an external tool called an isokinetic dynamometer. You strap somebody into it. We imagine, let's say it looks a lot like a leg extension in the gym. So we fasten our leg to it. We set via a computer the speed that we want the leg extension to take place. Regardless how hard we push against that lever arm, our leg will only extend at a constant speed. Very, very useful for testing, so we can see if there are deficiencies at certain angular speeds within a muscle. Very, very useful for rehabilitation, because we can really, really closely control how an injured body part is moving, but it doesn't really occur in real life. The last thing for us to think about today then are the various different roles that muscles can play during movement. So most movements are the result of several different muscles acting as a group rather than individual things going on. But they're not always playing sort of major roles in producing the movement. There can be underlying roles that have to take place in order for an overall coordinated muscle group action to occur. Most skeletal muscles are grouped into what we call antagonistic pairs. Okay? And these tend to be on opposite sides of the body controlling opposite actions. So muscles for flexion arranged in partnership with muscles for extension. Muscles for abduction arranged in partnership with muscles that cause adduction. So easy examples: biceps brachii, triceps brachii, quadriceps, hamstrings, pectoralis major, latissimus dorsi. So thinking about the opposite sides of the body is often an easy way to figure out the antagonistic pair. So within those opposing pair, one muscle takes on the role of agonist or prime mover. This is the muscle that is contracting to cause the key movement that we want to occur. At the same time, we have then the opposite muscle fulfilling the role of antagonistic. So this is the muscle that stretches or yields to allow that movement to occur. Okay, the text we use and the often way we describe it is the muscle relaxes. Right, it's a bit too simplistic to think about it that way because we'll often have some eccentric contraction during there to control the overall motion. But we'll talk more about that in class. So within a simple example, during a bicep curl, as we are lifting the muscle, biceps brachii is the agonist. It is contracting concentrically to cause the movement. Triceps brachii has to stretch, some degree of relaxation to get longer to allow the dumbbell to be lifted. Now, elbow flexion there is a key example. It is important to note that in a given joint, elbow, for example, and flexion, the agonist won't always be the same muscle. Various other factors can have an impact upon that. So last week we did mention in class the position of the hand, either pronated or supinated, will have an impact upon the biceps brachii's ability to perform work. So in the supinated position, palm up, because of the position of the insertion on the radius, biceps brachii can produce force and cause elbow flexion. If we then put that palm into a pronated position, so the palm facing down, we've therefore turned the radius and ulna. The line of pull isn't really efficient now for biceps brachii. So if we're going to flex the elbow now, a different muscle has to become prime mover. So in this case, it will be brachialis. So positioning of certain joints in relation to the one we're moving can have an impact upon which muscle is the prime mover in any given situation. The next key role a muscle can play is in a synergist. So synergists are muscles which sort of co-act at the same time as the agonist to support it in its key role. So there'll generally be a muscle that's very close to our agonist, performing a similar role, crossing the same joint, so it can support it and therefore reduce the amount of force that the agonist has to produce. So again, within a bicep curl type action, we have brachialis, brachioradialis that both also cross the elbow joint and can contribute to the elbow flexion. Okay, synergists may also play a key role in stabilizing sort of the other joints involved in the movement to allow for a more efficient movement to occur. And our final muscular role that can be played during a movement is a fixator. So a fixator tends to work statically to maintain posture in a certain position. More specifically, the fixator tends to stabilize the origin of our prime mover, okay, to prevent unneeded movement in that position. So, for example, again, if we take our bicep curl, the biceps brachii is going to be our prime mover to create elbow flexion. The origin of biceps brachii is up at the shoulder. So we will have fixator muscles stabilizing the shoulder joint to prevent it from moving during the bicep curl. So we'll have a number of muscles, deltoid for example, and then muscles around the scapula, such as the rhomboids, working to hold that in place. There may be a whole range of muscles, pectoralis major probably playing some role to stabilize the humerus, while biceps brachii is creating flexion at the elbow. So whole variety of muscles are in play at any given movement. Even a very, very simple movement there that we mentioned, like a bicep curl, which we are using to isolate biceps brachii. There are a number of other muscles occurring. And what we should also mention is during the lowering phase of that movement, the roles may completely change. All right. So the biceps brachii may no longer be the agonist. It all depends completely on situation and also position of various different body parts as to which muscle plays which role. And again, this is something we will look at in a variety of different examples in our second session later this week. A little reminder then, things to focus on prior to coming into college for your next session. We will begin with a look back at the names and locations of some of our key muscles. So make sure you refresh yourself on that. We will also do a number of exam questions on key points from this lecture that you've listened to this week. We'll then look at some of the key functional parts in a lot more depth and try to apply it to sport and exercise settings. So we will look at muscle fiber type and the characteristics of each fiber and which activities therefore they become best suited for. We will look at types of contraction, isotonic versus isometric, and the subdivisions of isotonic, and we will try to identify where those contractions occur in given movements. And then in probably those same given movements, we'll try to identify the roles that different muscles are playing, i.e. agonist, antagonist, synergist, fixator. So try to make sure you brush up on all of those areas prior to our next session. Okay, thanks and see you later this week.